am Talha Qaiser, a PhD student at Department of Computer Science, University of Warwick. And today I will talk about one of our recent work regarding digital tumor collagen proximity signature that predicts survival in diffuse large B-cell lymphoma. So lymphomas are malignancies that derive from lymphocytes, and they can be broadly classified into B-cell and T-cell lymphomas. And in this study, we have particularly investigated the role of collagen and tumor, and we have described a novel digital proximity signature and its significance in predicting overall survival in diffuse large B-cell lymphoma. So the proposed method is based on three main components, including cell detection and classification, collagen localization, and tumor collagen proximity analysis. Generally, collagen is regarded as an important component of acellular stromal microenvironment, and previous studies have shown that the presence of collagen is linked with patient outcome. But there are very limited number of studies that actually investigate the role of collagen in diffuse large B-cell lymphoma. There has been consensus that role of collagen varies with different types of cancer and their stages. And some studies have also found collagen as double-edged sword, so it may inhibit or promote tumor progression. But typically, the collagen acts as a barrier that resists tumor progression, whereas some studies have shown that the abundance of collagen may also link with tumor progression that may actually promote tumor. Initially, this study was performed on 32 patients, including 10 females and 22 male patients, with an age range between 24 and 90 years. After submitting this manuscript, we add another data set of 27 patients containing 9 females and 18 male patients with an age range between 30 and 86. In terms of cell detection and classification, we collected ground truth for 9 cases, and we have roughly around 30,000 patches with nearly 12,100 tumor cells and around 10,000 normal cell patches and approximately 8,300 patches from background regions. In first part of my talk, I will describe the algorithm for cell detection and classification. So here are some of the exemplar images from tumor and normal class. As we can see that some of the normal cells have weakly stained boundaries, and their nucleus substance have morphological resemblance with tumor cells. Overall, making this cell detection and classification a challenging task. So in order to solve this problem, we proposed a multitask learning framework that simultaneously predict cell class and location. The proposed framework process the given input and find and estimate the probability of each patch belongs to background, normal, or tumor. The other head of the proposed framework is responsible for predicting the central location of each cell. And it's also equipped with an attention layer which enables the cell detection module to precisely estimate the cell location. It is worth emphasizing here that most of the previously published approaches treated cell detection and classification as separate tasks, but over here we have proposed a joint framework that actually performs cell detection and classification. In terms of loss function, we have jointly optimized the weights for both these tasks. The first part of the loss function actually represents the log loss for true class and the predicted class, whereas the second part of the loss function actually computes the L1 norm between true class and the predicted class. Now, ideally, we don't want our model to learn any location information from background patches. In order to tackle this situation, we introduced a flag lambda that is only set to 1 for normal and tumor patches and 0 for background patches. During test, we use a multi-stage ensembling predictor. In the first stage, for an unseen image, we extract patches with different strides. And in the second stage, we train the same model on 3 different folds of the data set. So for each unseen patch, we get 3 locations and 3 classes, and each information is being fed into a function that estimates the probability map for each cell. And later we compute the average of these 3 predictions to conclude the final outcome. Here we have some of the qualitative results. As we can see that cells are phenotyped, normal cells are phenotyped as green, and tumor cells are phenotyped as red. And the image on the right-hand side has shown a more zoomed-in region of this left-hand image. Over here, we can see that model has not only estimated the tumor and normal cells, but it has also successfully identified the background collagen regions. Here we have a more zoomed-in region. We can see more details that how the proposed framework has identified tumor and normal cells and avoid the collagen regions. In terms of qualitative results, here we have the accuracy for each class. As we can see, that model has performed reasonably well for all three classes, but the overall accuracy of normal class is relatively less as compared to the other two classes, tumor and background. And that actually shows the variability within the normal class, mainly due to the presence of macrophages. In terms of comparative analysis, the proposed framework has achieved around 0.8 for F1 score. And as I said earlier, that most of the previously proposed approaches treated cell detection and classification as separate tasks, but over here we have combined both these tasks. And the results for comparative analysis also highlight the significance of attention layer that by the end helps the model in precisely predicting the cell location and overall improving the accuracy. In the last part of my talk, I will describe the algorithm for digital proximity signature. Now, once we have identified the tumor cells, the next task is to select a set of reference points from the collagen region. So later we can use that those reference points and tumor cells for proximity analysis. One simple way is to randomly sample the collagen points from the image. But the only pitfall of using such approach is that we may end up selecting a large number of reference points from one particular region of an image. So in order to avoid that situation, we first split the given image tile into small tiles. And for each tile, we perform stain separation and selected the DAB channel, perform the thresholding on the DAB channel. As we can see that the teal color actually showing the segmented collagen region. Once we have segmented the collagen region for a given patch, the next task is to select a single point from each small image patch that can later be used as a reference point of that collagen patch. So in order to achieve that task, we simply selected a single point which lies closest to the center of the patch, as can be seen by this blue dot. So after that, we compute the statistics around that reference points by simply counting the number of tumor cells lies within the vicinity of that selected point. And similarly, for each patch, we computed the frequency features. Once we have computed the frequency features for the entire data set, then we use simple yet effective k-mean clustering by selecting k equal to 4. And over here, we have specifically used Jessen-Shannon divergence. And we have empirically chosen Jessen-Shannon divergence to compute the distance between the centroid and the data points. So here are some of the qualitative results. As we can see that the proposed framework has highlighted the collagen region based on the density of tumor cells lies in the vicinity, in its vicinity. And this, the image on the right-hand side actually showing more details that how tumor and collagen are interacting. Similarly, if we scale the results on the whole slide image level, we can see a clear picture that how the interaction between tumor and collagen is undergoing in a given whole slide image. And in order to compute digital proximity signature, we simply compute the statistics as shown at the bottom by splitting the given results into 4 bins, by identifying it to very weak association, significant, and strong association. Now, from our results, we observed that a large part of a whole slide image actually contains normal regions. And in order to avoid the over-localization of very weak associations, we emphasize on weak, moderate, and strong associations. In terms of survival analysis, we can see that the patients with strong association between collagen and tumor produce statistically significant results for both validation cohort and for discovery cohort. In terms of Kaplan-Meier analysis, we can see that the proposed framework has stratified the patient into two groups, and our results suggest that patients with high tumor collagen proximity are likely to experience better survival as compared to other patients. And in conclusion, we have described a novel tumor collagen proximity signature that can predict overall survival in diffuse large B-cell lymphoma patients. Our results also show that the most important feature is the strong proximity between collagen and tumor cells, and that may be linked with better overall survival in diffuse large B-cell lymphoma patients. Further, we are exploring the utility of the proposed digital proximity signature as a predictor in larger cohorts of diffuse large B-cell lymphoma patients. Thank you.
Thank you very much. A clear presentation. This paper is now open for questions.
Very, very interesting. Have you looked at this or thought about looking at this in other tumor types, in particular breast?
No, at the moment we have just looked at diffuse large B-cell lymphoma, but as I said earlier that we are exploring it for larger, we intend to explore it for larger cohorts. So, I mean, we may explore it for different cancer types as well.
Because that sort of issue is very big in breast right now, so it seems like it could be quite interesting.
Yeah.
Okay, quite a small set of cases. Have you got plans to look at more cases with this technique?
Yes, when we submitted this paper, actually this was only conducted on 32 patients, but this is a pilot study. We then added another validation cohort, and in future we intend to extend our data set for larger cohorts.
So the DLBCL is, as a pathologist, it's a very wide dumping ground. So it has a lot of intermediate wide spectrum of disease. So the standard classification that we otherwise use as a prognostic is a germinal center type and activated B cell type. Have you done, tried to classify those in the two categories and then assess them separately? Or if you would be planning to do that in the next cohort that you're planning to study further?
Well, at the moment, we have not predicted the different types of lymphocyte. We have just emphasized on tumor and normal. But I think it's a very good suggestion to include that to see the effect of proximity signature if we actually classify different types of tumor cells, T cell or B cell lymphomas. So, I mean, it's a very good suggestion. Yeah.